Yeah, absolutely. So, the kind of the origin of the study was we know from work that predates ours that diabetes is a disease of obesity, of course, but also, principally caused by beta cell failure, the failure of these insulin producing beta cells in the pancreas. And one thing that had been clear in the literature for many years is that it's also considered a disease of proteotoxicity or protein misfolding. That these, abnormal proteins pile up, build up in the cell, and then that causes beta cells to die. Pretty much the field had really focused on only two proteins as like the key players in protein misfolding and diabetes. One being insulin itself or proinsulin, which is the form that is being processed and can misfold and lead to beta cell death, which is logical because beta cells make a lot of insulin. And then another protein that's called IAPP, which encodes a hormone called amylin that's co-secreted with insulin, and islet amyloid polypeptide, which is the protein name. It can also misfold in beta cells, and the misfolding of amyloid in beta cells leads to aggregates that also kill beta cells in diabetes, and not too dissimilar from the amyloid aggregates that you find in the brain in Alzheimer's. So those were pretty much the two players. And I had, one of the things we wanted to know were if we took a more holistic view, are those the only misfolded proteins in beta cells? Are there others? Where do those proteins live? What do they do to the beta cell? And so we did a relatively straightforward old school biochemistry technique where we could purify proteins that are insoluble. So proteins that misfold also become insoluble in solution. Um, and then we subjected the insoluble proteins to, high-end quantitative proteomics mass spec technology. And that's where this all started. We wanted to know where those proteins were. And it turned out the majority of those proteins, when we looked at cellular compartment, were in the mitochondria. Um, and so this kind of was really surprising to us. We didn't expect to find misfolded proteins there. We thought that we'd find them elsewhere, and we found them there. And that's what led us on this journey to ask why are the proteins in the mitochondria misfolded? What is the key regulator, and what does that do to health of beta cells? And what stood out to us was one of the key regulators of mitochondrial protein folding called LON protease or LONP1. It was highly enriched in the misfolded fraction, in the insoluble fraction, but the overall levels of LONP1 in beta cells were actually quite low. And the other regulators of mitochondrial protein folding, those weren't really that changed. It was really LONP that seemed to be one that was surprising to us that was pretty low in beta cells. And so that's what led to future studies where we knocked it out and beta cells began to die in rodent models, or we inhibited it in healthy human islets. Those beta cells began to die, began to have issues with secreting insulin properly. Yeah, so that's great. Great question. I mean, that was kind of the crux of the surprise of our initial observation from human type 2 diabetic islets. And then we kind of took to the molecular approaches to really say, okay, this isn't what we expected. We expect ER protein misfolding in beta cells, and we did find some proteins that were misfolded in the ER, but really this like heavy deposition of misfolded proteins in the mitochondria. I think the reason that we find this important is that, we're shifting the focus to an organelle that isn't getting the attention in this capacity. Most people think of mitochondria and beta cells. Yes, they don't work well in diabetes. Yes, they don't make enough energy, but we think of them as kind of what I often call a client, like organelle, like they're just there to supply energy and don't worry about them. And I think what our study kind of begins to reveal is No, when the quality control processes in this organelle break down and you're accumulating misfolded proteins there, this sets a cascade where the whole cell falls apart. Yes, you're obviously not getting energetics, you're affecting cell viability and health, but it's rooted in sickness in the mitochondria. It's not, oh, the mitos don't work well because things are going wrong elsewhere. Absolutely. Yeah. So LONP is a protease prototypically. And there has been some work that say that it also can function as a chaperone. Chaperones are, basically not too dissimilar from what you think of normally a chaperone. Like they're along for the ride. They're making sure things go properly, but they're also very important. Chaperones in the proteostasis context are important to help fold proteins and then also bind to misfolded proteins to clear them. And so it turns out that LONP works with mito HSP70, which is, one of three key, protein chaperone complexes that import proteins into the mitochondrial matrix. That's kind of the center, if you will, of the mitochondria. And that's where a lot of the proteins that make energy in the electron transport chain live. So the matrix is a really, important compartment. And so, what we were surprised to find was that we wanted to discern what role, because LONP has been shown to do so many things. We wanted to discern what its key function was during glucolipotoxic stress, right? So we're overloading these beta cells with nutrients. And so what we did is we overexpressed LONP. We also used a pharmacologic agonist of LONP. That agonist helped relieve beta cell death from glucolipotoxic stress, but it didn't tell us how LONP worked. So we also used a virus to overexpress a LONP mutant that lacked its protease activity by making a single amino acid substitution. So it can do all the other things LONP does, it just can't do the protease activity. It can't break down proteins. And overexpressing that mutant still recovered the beta cells from cell death after glucolipotoxic stress. So this suggests it's not its protease activity, it's protease independent. So where do we get to the chaperone function? So as I mentioned, other studies have shown that LONP binds and closely interacts with the mito HSP70 import complex to function in this chaperone activity and help fold proteins. So what we did is we rescued the cell death with glucolipotoxicity with the LONP overexpression virus. But the other thing that my postdoc did that was really elegant is she also put in this system then a pharmacologic inhibitor of mito HSP70. So if LONP is working through HSP70 in the chaperone capacity, that inhibitor should block the beneficial effects of LONP. And that's what it did. So this we used a couple of molecular approaches. One, mutating the protease away, the protease activity away, and then blocking the HSP70 to really resolve that the benefits of LONP are through its chaperone-like function. Yeah, I mean, that was what we only scratched the surface on with a single panel of this figure, but like we're eager to do. So we did use a relatively newly described LONP agonist that was, from a previous study in the literature. And when we put the LONP agonist on the glucolipotoxic beta cells, we improved beta cell survival. Now, that's in a culture dish. LONP does things in other tissues too. So I think one of the things that we're hoping to do is to do more studies with this agonist and see if you can give it systemically, will it have benefits in diabetic preclinical models, and then maybe we can go towards patients. Um, Are there ways to maybe further develop LONP agonists, using medicinal chemistry approaches to see if you can have, really safe, effective LONP agents that could be used for treatment of diabetes? I mean, I think that, our single, experiment in the islet, in the human islets, make us feel very hopeful. And then I think it's just up to us to kind of start developing these for in vivo experiments. Um, there have been other groups that have used the LONP agonists, I think, in models of kidney disease. That's, where we were inspired from the literature. So there must be some broad benefits for LONP in different model systems and some safety efficacy because it's been used in vivo and other models. So we're hopeful that we can translate it and use them safely in rodent models of diabetes and hopefully preclinical models of diabetes next. So, really, there were a few things that stood out to us. So, one is there's about 400 different genes associated with type 2 diabetes. Some of the genes we know how they work, some of them we don't. There are a few that are associated with mitochondrial function. We still need to know what those disease variants do to the function of these mitoproteins, and if they are modified, do they cause mitoprotein misfolding so that there's a genetic link to the misfolding that we see. LONP is, while we were really excited about our results, LONP is not a diabetes gene. So the dysfunction with LONP is from something else, some other genetic or maybe overuse issue in human type 2. So we're really keen to see what are those genetic links to protein misfolding, and then could we harness those for, as you asked about, personalized therapies. The other thing that stood out to us is that when we were working with non-diabetic beta cells or beta cells from rodent models that didn't have LONP deficiency, if you try to introduce toxic conditions in those systems, like glucolipotoxicity or overuse or overnutrition, normally the beta cells turn on a transcriptional nuclear encoded response to protein misfolding to help gird the cell up to clear those misfolded mitoproteins. But when we looked at the type 2 diabetic beta cells, that transcriptional response never came on. We don't know why. Did it like get burnt out? Is the transcriptional response a problem? Maybe part of the issue is that the cell never, like a type 2 diabetic, never turns on the proper response, and the proteins that normally misfold, all proteins misfold to some degree. That's kind of part of the kind of the garbage disposal components of cells, is like you got to clean up your messes. Type 2 diabetic beta cell, is that disposal, is the response mechanism not turned on? We don't understand that. And I think the next step is to understand, why is this response not being activated? Are there ways that we can promote activity of this response, of this transcriptional response to misfolded proteins, so that we can tell the cell, hey, there's misfolded proteins here, let's clear them. And then could we harness that therapeutically? I think that's another thing we're keen to learn more about. I think that, the most important thing for me is that this was the first, to our knowledge, study showing mitoprotein misfolding in type 2 diabetic beta cells. We're hoping this is the first of many. Like, we think this is a totally different way to view how beta cells fail in T2D, and that this is just scratching the surface of how we can, understand beta cell failure and then, learn, change our view of pathogenesis of this disease. Well, I'll just say that, we're really grateful for a few things in this study. One is our team at the University of Michigan and the University of Michigan Diabetes Research Center. We want to thank, the organ donors and their families. They're what make this research possible. And then funding from the NIH, the Breakthrough T1D, and the U.S. Department of Veterans Affairs really were important. I think in this era where funding is tight, I just want to make sure that we let people that watch this know that are taxpayers, like your work your tax dollars makes this possible so we can develop new therapies. It's more important now than ever.